Good morning, sir.
Hey,
Good
morning,
morning.
sir.
Good.
Good
It's good
morning.
afternoon, I think.
Good afternoon. Yeah.
Good afternoon over here.
Okay. Thank you, Dr. Pingle, sir. So, good noon and good morning, Dr. Sachin Korde. Okay. So, I, Saibra Boroste, welcome you all to the session second, day one of the conference. So, it's a great pleasure to introduce chairperson of the session, Dr. Milin Waksha. He is a professor of pharmaceutics and vice principal of MVP College of Pharmacy, Nasik. He did his B.Pharm from MVP College of Pharmacy in 1992, M.Pharm in Pharmaceutics from Birla Institute of Technology in 1994, PhD in Pharmaceutical Sciences from University of Pune in 2008. Sir joined academics as a lecturer in MVP College of Pharmacy in 1995. He is an approved PG and PhD guide. He has guided 60 M.Pharm students and 8 PhD students. He is the coordinator chairman Board of Studies in Pharmaceutics of Savitribai Phule Pune University. He is a member of BOS from 2005 till date. At present, he is a member of Faculty of Science and Technology of Savitribai Phule Pune University. He is a member of Senate of University of Pune, Grievance Redressal Committee and DTE, and a house leader of 20 teachers in Senate. Uh, Sir has published four patents. He has four patents to his credit and published more than 50 papers in international journals. His research area includes formulation of implants, test masking, ion exchange resins, and application of QBD for development of formulations and analytical methods. He has delivered several lectures and presentations in UGC AICT sponsored seminar workshops and refresher courses. He has secured research grants of more than 50 lakhs from various funding agencies. So, on behalf of Gokhale Education Society, Sir Dr. M. S. Gosai, College of Pharmacy, I welcome you, sir.
Thanks. Thanks a lot. Thank you.
Okay, sir. It's a privilege to introduce today's keynote speaker of the session, Dr. Sachin Korde. He is a regulatory consultant in London, UK. He is currently working as a regulatory consultant for various global pharmaceutical clients. Then he has research experience in research and development and technology transfer for 2.5 years. And he recently started the regulatory consultant career. He did his B.Pharm from YB Chavan College of Pharmacy, Aurangabad, M.Pharm from Pune College of Pharmacy. He did a diploma in intellectual property law from Bharati Vidyapeeth Pune, and he did his PhD from University of Bradford, UK. So, on behalf of Gokhale Education Society, Sir Dr. M. S. Gosavi College of Pharmaceutical Education and Research, I welcome you, sir. Today, Dr. Kode is going to present his expertise on topic novel abuse-resistant control drug delivery system, now I request him to proceed for the session. Thank you.
Thank you. Thank you, Saibra, for that kind introduction. Without taking further of your time, I'll start. Just a minute. Just open it and yeah. So you can see my screen now. Hello, can you see my screen?
Now, yes, we can see.
Yeah. Okay. Yeah. So good afternoon to everyone. Thank you for the kind introduction, Dr. Sahibra. Uh, 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 he gave my brief education background from India, and then after PhD, I did postdoc almost for 4 to 5 years, where I was associated with a few of my patented technology, which were at commercialization stage, and this is one of that technology. I would like to take you through this journey of development of abuse resistant technology. This is one of the most challenging formulation to develop in pharmaceutical research, and there were only one technology which was approved by US FDA, and they recently withdrawn it because it failed after introducing it to the market. So, what is novel abuse-resistant technology? So, uh, if you heard of opioid after cancer treatment, after surgical treatment, there are a lot of opioid drugs or like addictive drugs like heroin, ecstasy, and then oxycontin, oxycodone hydrochloride, codeine for that matter, which makes you addictive once you start taking it. So the resistance, abuse resistant term came into the picture. Can we develop a formulation which can help avoid any kind of deterring of the formulation? So many of the drug addict people, they do try to, crush the tablet, then put it in alcohol or put it in a mixer grinder, coffee grinder, to take the maximum dose and get a high out of it. So the basic two types of abuse resistant formulation are abuse resistant and abuse deterrent. The one example of abuse resistant is oxycontin, as I told you, which was the only product which was approved in US FDA, but they had to withdraw it recently due to the complication, and it failed the test of harsh, what do you say, pharmacokinetic in vitro analysis. The second example, abuse deterrent, where instead of using a physical barrier, they use some deterrent. Means if you try to misuse the formulation tablet or capsule, it will deter and hamper the actual dose of your opioid or abuse causing drug. These are the four other, main types by which we can achieve abuse resistance. First one is aversion. So with your main drug, there is additional drug which can give unpleasant effect. The second one is delivery systems, like you can develop different depot formulations or implants, uh, plastic polymers or high resistant polymers to which will avoid uh, immediate release into the body. Third one is the prodrug, which causes, if you try to tamper the formulation, it will cause a loss of activity, and a combination of these two or more than two. These are the main types of abuse resistant. The US FDA has classified these formulation in three types. So tier one is physicochemical barrier. You can coat your tablet or formulation. You can try to coat, or there are a lot of other options which can, like chewy polymers and stuff. But it's still challenging to optimize and manage the release profile of your drug. Tier 2 is block the effect of opioid upon manipulation. So as I told you, with the deterrent kind of additives, you can do that. And tier 3 is if you try to hamper, so the actual dose will not be delivered to the person who is consuming it. These are the main tiers of abuse-resistant formulations. These are the different schedule type of category, type 1, type 2 to type 5. The highest potential of abuse, this kind of drug, heroin, lysergic acid, diethylamide, and ecstasy. The second one is high potential for abuse. So most commonly used pain medication after surgical treatment or anti-cancer treatment is oxycontin, and then the current technology I have developed on oxycontin, oxycodone hydrochloride. You must have heard codeine was banned in India recently due to it is not that, what is a high potential or high risk drug, but it has been misused many times, and that's why it was regulated in India recently. So it is a type V. So you can imagine type 1 and 2, how potent these drugs are for abuse. To take your formulation of abuse resistant drug to next stage, you have very harsh pharmacokinetic or pre-marketing study you have to go through. The category 1 is like in vitro manipulation and extraction studies. In this in vitro manipulation, you have to try to crush your formulation, your tablet, your pellet, or your capsule. Try to crush it, try to break it, and then try to grind it, and then put it in alcohol, put it in vegetable oil, put it in boiling water to just make sure to just cross check will it release the drug more than what it is needed for that patient or for that person's treatment. Category 2 is pharmacokinetic studies, the dissolution studies. I'll take you through the what are the important pharmacokinetic studies we have to do, and then clinical abuse potential studies. So the Oxycontin by Purdue Pharma, it passed category 3 clinical abuse potential studies, but after launching into the market within 1 year, it's a big lawsuit on them, and USFDA has filed a case against them for almost $200 million. So, I mean, those of you who will go ahead and do research or involve yourself in research activities in pharma industry, this is one of the most challenging technology or platform you can come across, and then you can try to pursue. As there is not a single product in the world at the moment for this kind of technology. So this is a bit old data. So the top players are like Purdue Pharma, as I told you, Johnson and Johnson, Pfizer, other generics. I don't think so. Any Indian pharmaceutical company is involved in abuse-resistant formulation development as such. The trend for the narcotic drugs and oxycodone, it is keep on going on increasing really, for all the pains of post-operative surgery, and then anti-cancer, most of the anti-cancer treatment where you have to surgically remove the part and then follow up with the chemotherapy. These are the market players. So Oxytrex, as I told you, Pen Therapeutics, King Pharmaceuticals, and Acura Pharmaceuticals. But none of them, they're in phase 3, but none of them has, achieved the stage to reach to market yet. The technology in the market at the moment, the Oxycontin, the Purdue Pharma, which I discussed recently, it's a hard coating on your drug. It's a kind of tablet. It's a white tablet coated with the hard crush-resistant polymer. So if you try to crush it, you won't be able to crush it. The second type or second types of polymer or technology which other market players are trying is gummy pill. Like some if you try to put it in water, it will give a gum like consistency, and it will be difficult to extract the drug out of it. Some of the companies, they're trying different approach using antagonist of your drug, so that if you try to tamper the formulation, it will lose its activity. And then the fourth one is like if you try to take, which is more than needed, there will be some additives which will make you sick, different colors or smell if you try to extract the drug. This is Navipro database from USFDA, and then these are the technologies which are hopefully from last 4 or 5 years they're in pipeline, but none of them reached market yet. So these are just example. So you can see, I mean, there is huge interest in this technology, and even company like Pfizer couldn't get, even Reckitt Benckiser couldn't get this formulation out there in market yet. These are the patents, just for your reference, which are filed on the abuse-resistant technology. If you Google patents, there are a few in 2021 and 2020 at very early stage, but there's no further progress really. So the technology I have developed at University of Bradford, it's tier 1, so it's resistant to crushing, and then by any means you are not able to crush it or tamper it to release more than 15% of drug. So that's how I optimized it. It took us a lot of trial, a lot of optimization, a lot of different combinations of your additives, excipients, polymers, and there is this flexibility. So dosage can be adjusted, and then the attempt was to develop it into 3 or 4 different formulation to just maximize the application potential. In an oral route, we have optimized it for a pellet and tablet form, and then for transdermal delivery also, we try to optimize it in the form of microneedles. We found out that for this technology, not only abuse resistant, but you must have heard of alcohol dose dumping, Paladon, paracetamol, Paladon, which causes, if you consume alcohol and then take the Paladon, it causes a dose dumping, and which is not good for the purpose you're taking it. So this technology can be applied for alcohol dose dumping or the drugs which are prone to alcohol dose dumping. The first formulation, as I told you, is pellets. So this is how it looks, product. We can put this in a capsule. We can try to put it in capsule form or in like a unit dosage form. So this is before and after dissolution. So we called it a cage, really, our cage technology. If you can see before and after dissolution, it kind of opens up and then releases all the drug and remains its intact structure after releasing the drug. So we have optimized it for like more than 5 or 10 times trial. We usually do in triplicate, but we did it in like 9 to 10 times repetition result to just make sure that we are getting the exact results. So to pharmacokinetics or in vitro studies, you have to do. So we compared our data with the US patent, which is on the right side of the table, with our technology. So you have to do initial release in 0.1 normal HCl, 40% alcohol, vegetable cooking oil, and then boil and cool, like boiled water. And then, as you can see, it shouldn't release more than 15 to 20% of your drug in any of these trials you are doing for the in vitro analysis. So we were in a position, if you compared with the patented data here, there is a US patent. Our technology is far better than that US patent technology. The second formulation was injection molded tablets. We here at University of Bradford, we have excellent facilities in terms of extrusion and injection molding and micro molding. So this is the machine we used, Fanuc RoboShot, for the injection molding tablet preparation. So this is we prepared. I prepared 3 different types of tablets to just have a backup if one of these tablet fails in any kinds of pharmacokinetic trials or in vitro trials. This is in vitro dissolution. This is how the tablet looks after dissolution. So it just releases the drug and holds its a cage or intact structure after dissolution also. This is the similar comparative table for type A, B, and C tablets, injection molded tablets, and you can see the release pattern. It's way low, way low. So the first, when we drafted the patent draft, we were supposed to file a pellet formulation, which I showed you earlier, and then we decided that we can cover and try to maximize the application potential and try to cover injection molded tablets and microneedles as well. So this is a crushing strength. If you can see tablet after breaking, it takes more than 450 Newton force to break the tablet. But after breaking also, if you put those pellets, broken pellets in the dissolution media, and if you try to do all those studies in 40% alcohol, boiled and cool water, 0.1 normal HCl, or for that matter, vegetable oil. Even we try to do a study in fruit juice, orange juice and stuff, because these drug addict people, they tend to try various stuff. If you have seen in the movies or for your reference in any YouTube videos, or they try to take it to the spoon, and then with the lighter they try to boil that water in the spoon and try to dissolve or extract the drug in the spoon, and then they inject it to themselves. But we did all those studies, and then by any means we were able to achieve that our formulation is not releasing the overdose or like the dose more than needed. The third formulation was microneedles, which was very challenging, and then bypassing microneedle as the dose is very low, and we had to make sure if they try to tamper or break the microneedle, so try to extract it by any means, it shouldn't release that drug. So this was the most challenging formulation optimization out of all three. So we use Battenfield micromolding facility. The attempt here was biphasic microneedles, biodegradable microneedles. We tried and optimized a lot of biodegradable polymers to optimize this drug, this formulation. This is how the microneedle looked. If you look at the length and the what do you say evenness of the microneedle shaft throughout the length, it was really good. And then if we have any experience in this kind of technology, it is very challenging if you are not using the ideal polymers to optimize microneedle formulation or dosage form. So this is confocal imaging, like how much was the uh, drug loading and how it appeared after the dissolution or after transdermal studies. We used human skin and animal skin to do the diffusion studies and then check the release optimization of the formulation. So this is the shingle shaft, how it looks in confocal single microneedle imaging. So this is a bit confusing, what do you say, table. And then I would say, so these are just size, drug loading, and mechanical strength of the microneedle we prepared. And we were able to load 10% of the total weight of the microneedle, which is quite commendable. We tried to load to the 30% of its capacity of total weight, and we were able to achieve it. So I would like to say, I mean, the formulation we developed, I mean, there are a lot of great interest shown from USA companies, and it's this technology is with University of Bradford, and then it is at various levels of commercialization. It has long way to go because the Purdue Pharma, after approval from US FDA, it failed miserably, and so there is huge risk, and it will need very harsh testing and clinical optimization again before it reaches the market. But I was in a position to, what do you say, to achieve excellent abuse resistant. There's a dosage flexibility and excellent inventory performance, and then this technology is ready to tech transfer. I mean, when we speak from taking technology to from pilot scale to large scale or scale potential, uh, there are a lot of challenges to it, but the technology we have used, I mean, it is possible to take it to the scale up level or do the mass production around 100 kilogram or 1 ton a day. Thank you. I would like to take any questions if you have.
If there are any questions, you can ask. Dr. Pingay, are you there? Dr. Korde, I have a question, like
Sure.
just trying to understand what you have been doing.
Yeah.
This, I mean, Purdue's example and the fine imposed, I would say, was a kind of deterrent maybe for others to come in.
Yeah.
And as you had said it is quite risky also. But then, because there are no, I mean, very few players in the market,
Yeah.
it also has a big potential. Like,
Yeah.
and the product that you have optimized and developed, you
Yeah.
feel to be very confident that it will pass
Yes.
the USFDA requirements. Of
Yes.
course, the scale up is needed for.
Yeah. Yeah.
I mean, those things. Scale up is yet. I mean,
I
those
mean,
studies are.
we did trials up to 10 kilogram at our lab scale. Yeah. So, and we are sure, looking at the 10 kilogram batch, the results and characterization we did, it is possible to take it to 50 kilogram or 100 kilogram at this moment. The second thing is only big players can take this kind of risk. I mean, Purdue Pharma, they earned like 300, 400, almost $500 million during that first 1 year when they introduced into the
Ah,
market after US FDA approval. But it was a big fine, like $200 million pound, dollar fine. And only, I mean, the current, I would say Pfizer and J Johnson and Johnson will be the one of these two will be in the market
go
very
for it.
soon, in next, I would say, couple of years maybe. So there were 2-3 companies. I mean, it is confidential. So they have approached University of Bradford for this technology,
Okay, okay.
and then the data transfer and their uh, as this technology is very, what do you say, very sensitive to do to transfer as well. So they're in different agreements now. So
Right.
yeah,
But then, like, I was a bit surprised. Like, I mean, the product was developed by Purdue and it was approved by the US FDA, and then
yes,
it
yes,
was withdrawn. So the
yes.
whole onus was on Purdue. Like, FDA didn't, because it was FDA who
Give
approved
the
it.
permission, yeah, but the sole, what do you say, ownership or the formulation, they had big investors on their part, Purdue Pharma, to get the approval for Oxycontin. But
Okay.
yeah, they knew earlier, I think. There was a news that they knew that it is failing. People are still tampering it, drug addict people or whoever
So
on Oxycontin.
then the FDA criteria were not that rigorous, you can say.
No, they did. They informed them earlier. They tried to bypass it
Okay,
by
okay, okay,
other routes, and so they got caught at the end.
okay,
And
okay,
yeah,
okay.
yeah. This happened recently, I think, early this year. Yeah,
Okay, okay.
2021. Yeah.
And the market mainly is US market, or
So
it's like
majority of abuse, it happens.
low
It's not
prevalence.
correct to say, but it's 70 to 80% in USA, and
Right,
then some Europe countries in Europe, Canada, and then
right.
China. Yeah.
But
Yes,
then
Dr.
China also, like,
Sachin, you have one question from delegates, Shraddha Patil.
Yeah.
As you discussed in one formulation, coating is hard in tablet.
Yeah.
How it will be eliminated from body?
Yeah, so the release timeline for the formulation, it's almost, we studied for 12-hour to 18-hour, you need a constant dosage. So I just showed the representative example for the tablet. It remains intact until the drug is released, but it dissolves later on. So it will be, it has biodegradable nature, and it will be like other tablets. It will be consumed in the body. So it won't remain intact until it gets excreted from the body. I have hope that answers the question. Yeah.
Yes, yes, Dr. Sachin, thank you very much. Uh, on behalf of my college, Vocal Education Societies, the Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I am thankful to Dr. Sachin Korde, session speaker of session 2 of first day of international conference. I am thankful to Dr. Milin Waksha, who is chairperson of this session. Dr. Sachin, after listening your session, I remember a quote of Paulo Coelho, who is a Brazilian
Yeah.
novelist.
Yeah.
An enthusiastic heart finds opportunity everywhere. Such kind of opportunities young and dynamic researchers should find. You already found an opportunity and you got the fruits of that opportunity also.
Yeah.
So thank you very much. Thank you, Dr. Sachin, for your precious talk in our conference. On behalf of Vocal Education Society, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I am thankful to Dr. Milin Wag, sir, Professor and Vice Principal, MBBS College of Pharmacy, Nashik. Sir, I am very much thankful to you for chairing a session at conference of this our college. Okay.
I would like to
Yes.
share something.
Just a
Uh,
minute, sir.
yeah.
Okay, okay.
So,
I request Dr. Milin Wag, sir, to share his concluding remark of this session. Please, sir.
can I
In
say
fact, I
something?
think,
Just 1 minute. Can I say
ha,
something? Sorry to interrupt here.
please go ahead, Dr. Korde.
I mean, I have fond memory with Milin Waksha. I'm
Okay.
sure you might not remember it. You did your PhD while in 2008, Pune College of Pharmacy.
Right.
on fifth floor chemistry. So you
Yes,
used
yes.
my laptop. I
Okay.
was doing my masters that time, and I have a, yeah. So very fun.
It's a small world. We could keep on meeting.
Yeah, yeah. So yeah, I would like
Dr.
to share
Yadav
this.
was the external, maybe other professor
Yeah,
was there. Right,
yeah,
right. So great coincidence. We are
yeah.
meeting again today. Dr. Prashant,
Yes, sir.
should I
Sir,
should I
please conclude.
summarize?
No, sir. I want to say thanks for both the chairperson and session chair. Before that, if you want to conclude this session, you please say a few words about this session, please, sir.
In fact, in between, I mean, after his speech, I had some questions and I asked him, and I found this very, mm, I mean, a different area to which we in India are not exposed much.
Yeah,
Maybe abuse is not that prevalent, or maybe that recognized or monitored in
yeah,
India. But that is going to be there. Just a recent case was there in, I think, in India, a lot of drugs were found. So it is something that is under the carpet. A lot of things are happening, I think, in India.
yeah,
So this is very relevant in the Indian context also, at least to have a glimpse of like what the world is thinking about,
yeah,
and how. I mean, it was very interesting, like how you can have a, drug delivery system itself to stop the abuse of these drugs.
yeah,
So it was nice session, and what was more interesting is he shared his own work in Bradford, which is almost on the stage of scale up and then
yeah,
commercialization.
yeah,
So that is something that is very interesting as well as inspiring, and I must say, quite an achievement at this young age.
thank
So thanks, doctor, for I would say opening up a window to this area. I enjoyed and learned from this session, and I hope all the other participants also learned a lot from this. So, this is where I summarize today's talk by Dr. Sachin Korde. And personally, I thank Dr. Management of Dr. Sir M.S. Gosavi College of Pharmacy, the principal, as well as Dr. Sunil Amritkar, as well as Dr. Prashant Pingay for inviting me, and Saibra Boraste for the coordination. Thanks a lot. Over to you.
Thank you, sir. Thank you very much. On behalf of my college, Gokhale Education Society, Sir Dr. M.S. Goswami College of Pharmaceutical Education and Research, I am thankful to Dr. M.P. Waksha, sir. He chaired a session. Thank you, sir.
Thank you.
I am thankful to Dr. Sachin Korde, Regulatory Consultant, London, UK. Thank you, Sachin sir, for your precious words and valuable talk in this conference. I am thankful to you on behalf of my college and organizing team of our conference. Thank you very much, Dr. Sachin.
Thank you.
thank you so much.
So, with this, I would like to say thanks to Milin Waksha and Sachin Korde sir for your
Thank you.
valuable time. You spare about 1 hour for us. Thank you very much, sir.
Thank
Please.
you,
Thank you.
thank you
Have a
for the
nice,
invitation.
Thank
have
you.
a
Thank you to
nice
Thank
day.
you.
Saibra. Thank you to Dr. Sunil.
Delegates, you're going to join next session in next 2 to 3 minutes only. The session will be by Dr. Sanjula Babuta and the chairperson will be Dr. Vandana Patrawade. Thank you very much.
Thank you, thank you.